Our next speaker is Meleah Hickman, Ploidy tug-of-war. Evolutionary and genetic environments influence the rate of ploidy drive in a human fungal pathogen. A non-Saccharomyces talk, I guess. We'll allow at least one of those.
Yes, I'd like to thank the organizers for having a non-model system represented here, but yeast nonetheless. So I'm going to start off and just begin by saying that different budding yeast species across the fungal kingdom have a very clear correlation in their ploidy, that is the number of chromosome sets that they contain in the genome, and their cell size. So haploid cells are smaller than diploid cells, and diploid cells are smaller than tetraploid or polyploid cells. And we see this in the sort of premier model system, Saccharomyces cerevisiae, which Bob Mortimer showed in the late 50s, very clearly that haploids are smaller than diploids, are smaller than tetraploids. But we also see it in the pathogenic fungi, such as the Basidiomycete Cryptococcus neoformans, which is, my mouse is still not working, I apologize for that, Cryptococcus neoformans, which causes, we inhale their spores and it gets into our blood-brain barrier and causes meningitis, and also the fungal pathogen Candida albicans, which is more closely related to Saccharomyces cerevisiae and the system that I'm going to be talking with you primarily about today. So just for those who don't think about Candida albicans or even other non-Saccharomyces species, I'm going to give you a little brief background. So it's been studied since the mid-1800s. It is a normal inhabitant of our GI and GU tracts. I'm sorry, I'm really loud. And despite it being a normal commensal inhabitant, it's actually also the leading cause of fungal disease. And this disease ranges from superficial infections in healthy individuals, that's thrush or yeast infections in females, to much more serious systemic bloodstream infections in people who have compromised immune systems. And that can sometimes result in mortality. Sort of exasperating that problem is that there's a very limited number of antifungal drugs available to treat fungal infections, and that resistance can arise very, very rapidly. And one mechanism of this resistance is actually changes in chromosome copy number. And so about 50% of clinical isolates that have been shown to be drug resistant actually carry some sort of aneuploidy within their genome. Not only that, there's a wide range of phenotypic variation in Candida, and not only does it grow as a budding yeast, but it can also grow as pseudohyphae or filamentously and form biofilms. There's also a large degree of protein variation because of a CUG codon change in the Candida clade, whereby CUG is translated as serine about 97% of the time in place of leucine. And then finally, sort of the topic of today is that for a very, very long time, historically, Candida albicans was exclusively described as an asexual and obligate diploid organism. And I should add that it's also a very highly heterozygous diploid organism. And so I'm going to sort of shoot that to shreds. I don't expect you to trust my word on it, and I'm going to show you that if we take a survey of Candida strains, and just look for their DNA content. So survey meaning laboratory strains or strains that we expose to external stress or passage through a mammalian host, or even strains that we acquired from people from clinics. You can see that by and large, these isolates are indeed diploid. And then if you look at the clinical distribution of ploidy range, that is extended, and that gets exacerbated if we look at the outliers from these data sets. And so isolates that can deviate from the diploid state can deviate quite dramatically. And we see this even, there was work published just last month on ploidy variation seen in Saccharomyces clinical isolates as well. So it's not just consistent with Candida, but other budding yeast species as well. And so the question becomes, how does the environment either promote or maintain these non-standard ploidy states? And so I just want to revisit this nutrient limitation hypothesis that was posited by Lewis in the mid-80s that would say that it actually has nothing to do with genetics whatsoever, which sort of rings false to a geneticist's ears, but rather that it has a lot to do with physiology, and that the haploids, because they're so small and have a very high surface area to volume ratio, under nutrient depletion or scarcity are actually going to grow much better because they're going to be more efficient at bringing in nutrients, and so they will have an advantage when nutrients are scarce. And so we wanted to test that in Candida albicans, and we did this by using 3 sets of strains that varied in their ploidy and in their genetic backgrounds. And so the first set that I'm going to talk about, I'm going to call the heterozygous genomes. And so we have a diploid and 2 tetraploid strains. These are representative of the laboratory reference strain. So the tetraploids are either derived from a mating or mitotic defects to generate tetraploids of that same genome. The second set I'm going to call homozygous genome. So that is a haploid strain and its homozygous diploid derivative. And then a second haploid strain that is not isogenic, but is actually very similarly composed of in terms of its alleles. And then finally, a third set that is a pair of clinical isolates that came from the exact same patient, but are temporally spaced. And one is a diploid and one is a tetraploid. And they are similar genetically, but they are different from the laboratory reference strain. And so we now have the ability to dissect the interactions between ploidy, allelic heterozygosity, and growth environment. And so if we test just simply, are there changes or differences in growth rate under complete or replete media, you can see that we don't actually have much difference between diploid and tetraploid strains. So the letters indicate non-statistical significance between data points. But there is a very severe reduction in fitness in the haploid and homozygous genomes, suggesting that heterozygosity is actually contributing to fitness more so than ploidy is. But what about if you deplete the nutrients available to these cells? Will that hold true, or will haploids now have the advantage? And what I'm showing you here are 3 different growth conditions where we've either reduced the amino acids, reduced phosphate, or reduced nitrogen. And what you can see is that in none of these cases do haploids ever win in terms of the fitness game. But that it becomes much muddier to see who wins at all. That sort of a lot of the strains are behaving either similarly or not distinct enough to make any clear predictions on ploidy or genetic background on their growth abilities in these environments. Okay, so while I've shown you that there's clear and extensive ploidy variation in Candida albicans, there's an observation that we've seen several times now where if you propagate these strains in the lab for extended periods of time, you will get this convergence towards diploidy. So haploids, if tetraploids start off and we just continually serially propagate them in the lab, they will fall down to triploid or diploid through these aneuploid intermediates, and that was published last year. And haploids, if you propagate them under rich media conditions, will generate mitotic defects and generate diploids sort of arising under continuous culture, and that was published a couple of years ago. And we see the same sort of observation in Saccharomyces also. And so this is work done by Elisa Geerstein when she was a graduate student in Sally Otto's lab, where you have haploids, diploids, and tetraploids all converging over much longer time periods back down to the diploid state. And so this comes to this sort of idea of ploidy drive, and that is that there is some force that is acting to push an organism back down to its baseline or historical ploidy. And the question we then asked was whether or not the growth environment might influence this ploidy drive at all, and what the interactions are between the environment, the genetic background, and the ploidy, and this sort of tug-of-war environment. And here I have this sort of like lovely sunny day, nice even playing field, but you can imagine that if the ground itself was like on a mountain, that the two parties on either side of the rope might not actually be equally weighted, or if the weather, whatnot, you're in some sort of hailstorm or whatnot, might be have a different outcome. And so we took these same sets of strains, the homozygous strains, the heterozygous lab strains, and the clinical strains, and then propagated them for about 150 generations in serial batch culture and under these different media conditions with 12 replicates per strain per condition, and then measured the change in growth rate as well as the change in ploidy for all of these lines. And what we see is that we can indeed sometimes observe, even within 150 generations, adaptation to growth environments. So here, the homozygous genomes, which were growing really, really slowly under complete media, now grow much faster. But whereas in reduced amino acids, phosphate depletion or nitrogen depletion, you don't get this sort of conserved everybody adapts to the same degree or at the same rate or at all. And so there's, again, sort of no clear predictor of who's going to adapt or how well. Now, if we look at their ploidy, and so this is just the average ploidy change across the group of replicates per condition, and I've shaded out the sort of diploid range in gray so you can orient it. So you're looking at the arrows are indicating changes in ploidy either upwards or downwards. So under complete media, haploids bounce up to a diploid state, and tetraploids sort of fall down to diploid, that idea of ploidy drive converging on diploidy. But that's not what we see under minimal amino acids or under phosphate depletion, where the haploids actually stay haploid even after extended passaging under these growth conditions, although we do see ploidy reduction in tetraploid strains. And then finally, under nitrogen depletion, we see something very similar to complete media, where the haploids go up to diploid and the tetraploids, some of the tetraploids go back down towards diploidy. So if we now look and compare, does the change in growth rate correlate at all with the change in ploidy? Are those two things connected or not? We can start to sort of see what the relative strengths are of each component. And so what I'm going to show you is this graph where we've plotted the change in ploidy along the x-axis. And so things on the right-hand side are gaining in genome size. These are mostly going to be the haploid isolates that are increasing their DNA content. Whereas things on the left-hand side of the graph are losing DNA content. These are going to be the tetraploid strains as they fall back down to tetraploid. And then on the y-axis is the change in growth rate. And so things very high up on the graph are going to be the lines that have had growth rate increases, and things that are much closer to zero are going to just be reflective of strains that have a very high level of genome instability but do not reflect any sort of selection for a higher growth rate. Okay. So when we look at complete media, you can actually see that the majority of the haploid lines have indeed not only increased in ploidy, but also in their growth rate. And the tetraploid lines have either decreased in ploidy without a subsequent increase in growth rate, or haven't changed in ploidy much at all. And so it seems that under complete media, there is some sort of force driving towards larger genomes. Which is in contrast to what we see for reduced or minimal amino acids, where the haploids are maintaining their haploid state, and the tetraploids are losing DNA content, and as well as gaining increases in their growth rate. And we see something similar to that under phosphate depletion, where the tetraploids, by and large, are losing their DNA content and gaining increases in their growth rate, whereas most of the haploids, but not all of them, are maintaining their haploid state while not even really making any gains in their fitness. Although this is a less clean picture, you can see that there are some individual replicates within this experiment that do show gains in ploidy in this condition. And then under nitrogen depletion, we actually see this sort of, again, this convergence towards diploidy, with the haploids getting bigger and the tetraploids getting smaller, and they also have corresponding gains in fitness associated with those ploidy changes. Okay, so in conclusion, I hope to have convinced you that Candida albicans can display extensive ploidy variation, and that this nutrient limitation hypothesis is not well supported. And it should be said that there are lots of other studies in Saccharomyces and other budding yeast species that also do not support this idea of a nutrient limitation hypothesis promoting haploidy. But rather, in Candida, it's the allelic heterozygosity that really confers very large fitness advantages under most of these conditions, as opposed to ploidy per se. That ploidy drive is this convergence to the baseline ploidy, and can be, but doesn't always have to be, independent of selection for faster growth rates. And then finally, that non-diploid states can be maintained under particular growth environments, and that was sort of indicated with the haploids under reduced amino acids or phosphate limitation, where they maintained their haploid state despite not necessarily growing very well in it. And so with that, I would like to acknowledge my budding lab at Emory University. I'm a new faculty member there, and so all of the members there. But by and large, this work was done in collaboration with a fellow postdoc at the University of Minnesota, Eliza Gerstein, worked with Judy Berman, and an undergraduate, Susie Lim. And with that, I'd like to thank you for your attention.
So my question is that, as we all know that the TOR pathway, the target of rapamycin pathway, is required for the growth rate, right? And the TOR is also responding to the environmental nutritional status. So I was just wondering whether the TOR levels changed during these different haploid, diploid, and the ploidy. Thank you.
Yeah, that's a great question. We did not look at all at TOR or what would rapamycin, affect this at all.
Well, I was wondering, have you done RNA-seq to look at allele-specific expression, especially in the heterozygous situation?
We have not.
Because that would obviously get you mechanistic
Yeah. No, that's
stuff, yeah.
on the list.
All right, thank you very much, Meleah.